Hi there. One of the last videos for the metagenomics part of Introduction to Bioinformatics at Utrecht University. We're annotating genomes this time in assignment M11, annotation. So for annotation, the process of annotation, just to define it, is to take, in our case, a FASTA file with a genome sequence, not one linear or circularized genome sequence, but many contigs that together are a draft genome or a metagenome assembled genome. And within these strings of ACTG, we're looking for more meaningful information in a way. We're gonna annotate it and say at this position starts a gene, and at this position it stops, and then the position a bit further on there's another gene, etc. And all that information can be found in the genome. That's a long process, so we're making an annotation next to it, so to say, this is where this gene is and we think it has this function. Okay, so let's see how we have to do that. Make a directory for the prokka output. So prokka is the program that we use to annotate. It's like checkm, it's a whole workflow of many programs and many steps that perform this task in an automated way. So we're going to make their data / annotation, and I'm picking bin1 to annotate. It cannot make two directories at once, so I can do first this and then this, or I just type -p as an option, and then it also works. Prokka, how does that work? There's some error messages, that's not a good sign, but let's just ignore that for a moment. Prokka synopsis, that's nice. Rapid bacterial genome annotation. Usage, prokka options context.fasta input. So I think I put it in the description here. Yeah, so we are gonna definitely use these options because prokka doesn't like the names of our contigs. Oh, they're already here. So that's where they copied. The path to your bin, let's put that in. Data /bins /bin.1. Use tab to autocomplete. The output directory, let's make that data /annotation /bin1, the folder we just made. I don't think we need other options. Nope, there we go. So it's finished way too fast. Let's see what's wrong. The sequence databases have not been indexed. Please set up prokka. Please run prokka setup db first. Okay, I don't mind. Let's do that. It seems we are in trouble. So this server does not have the dependencies necessary to run prokka. We need to make BLAS DB 2.8 or higher, and we don't have it. So since I'm filming this last minute with the whole coronavirus issues, I'm not going to manage to install that before the start of the course. So again, that's a bit of a bummer at the end of the practical, but I'll show you how it works on my own computer like I did with checkm, and I'll be absolutely sure to get you the output file so we can do the last steps with all of us by ourselves. Okay, so here we have again the bigger server I mentioned in the other video where we ran checkm. This is where we also have prokka installed with all databases and proper dependencies, etc. So I'm in the data folder. I'm doing just the same as we did a second ago. Bin /bin1.va, and the output. Oh, I have to make that one. Make their annotation /bin1 -p. Yep. Prokka bins bin1.va, annotation bin1, Now I will just copy these options from here and paste them in here. Oi, that's not supposed to happen. Remove this nonsense. That looks clean. We run it. Oh, come on. Let's troubleshoot live. Why not? Oh, I'm actually messing stuff up. Out there, no, out there, there it is. But did I put that in here? No, I didn't, so that's what's wrong. Out there. So I have to actually remove that annotation bin1. Some programs demand these directories to be non-existent. By making these directories themselves, they know for sure that they're not overwriting anything that they shouldn't be overwriting. So I guess that was a bit real-life troubleshooting that happens to anyone as completely normal. But if you read error messages carefully, often these programs give you nice instructions and you can solve these things by yourself. I'll let this run for a second. Okay, prokka is done. I figured I would make a loop with all the bins. So since you guys have made loops 3 times now at the very least, if you have done everything in the practical, I'll show you what I did. So let's see. The first command we ran was like this. Prokka bin1, put it in this directory, the options, etc. And what I figured, I also want to do bin226, so I said bins, an array, that's I call bins, is an array of 5 elements, namely 2, 3, 4, 5 and 6. Then I made a loop, and the loop looks like this. Doesn't format very nice, but you get it, I guess. So for the variable b in the array bins, like we've done before, do prokka, just the same as we did before. I don't think that selecting it actually helps. So do prokka bins, and then the number I replace with the variable, mind the double quotes here, out there, annotation, bin, and then again the variable b. So this will be bin 2, bin 3, bin, etc. I give it some extra CPUs because I'm impatient, and we move on. So that's what I did. And let's look in the folder annotation, and there is 6 folders. Perfect. I want to see what's in all of these folders, and there is a whole bunch of files. Yeah, so I'm gonna copy these files onto the CoCalc server, and then we continue on from there. Okay, so the files are on the server. I put them in the data/annotation folder, and you can see all 6 directories here that we just made. What was it in here? So back to the CoCalc server. Now let's have a look at what all these different directories contain. So there's a bunch of files, really a lot. So I can show you a few. FAA is often amino acid FASTA, NA is often nucleotide FASTA, GBK is a GenBank file, a log speaks for itself. GFF, that's the file that we're going to use later. Table, tab separated, text, a whole bunch of files. So I made this annotation for all bins. You only need one. Pick the one you like based on the table or a hunch or whatever. So let's have a look at what this input is. Yeah, let's just play with these bits. Data / annotation. I'm gonna do bin 1 and what's in there? .faa. Okay, so this is a FASTA file. You can see from the header here. It has some code, hypothetical protein, that doesn't tell us much. And it's clearly a protein sequence. Now let's look for FNA. Oops, that's not what I want. FNA, there we go. So here you have again some code. It's a FASTA file, but this time in the DNA. What's in there? Let's look at the error. Discrepancy report results. Okay, I'll just believe that for now. FFN. So this is the nucleotide sequence of the proteins. What's then the difference between FAA, FNA? Huh. I actually don't know. Maybe I'll come back to that later. FSA, is this the entire, oh, it could be the entire, so the entire bin is copied in there. GBK, so all these files, this looks very, so this is some kind of table or some kind of other format. And the one we want, I'm going to open now, is the GFF file. So in this case, I want to remove everything. Grep. I want to remove everything that has this hashtag in front. So what I'll do is so we've used grep before, right? So grep filters all lines with a hashtag, but then I use grep -v to have all lines without a hashtag of this file, and then I have the head again of that file. There we go. Like always, it is a very big table. Maybe you recognize this just from the other file. So this is some code that these genes get. Then there is protocol, that's actually the program that finds the open reading frames. CDS is short for coding sequence. I guess this is the start and the stop. There's some numbers. There's an ID, parent, blah, 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 blah. So here's some more information. Prediction made with protocol. Locus tag is this, okay. So that's all more from computers to read than for humans. And actually there's even a product. So this gene makes a hypothetical protein like we've seen before. So there's one thing that I find particularly interesting, that's the ID here. So this is, no wait, that's not the one I want. Where is it? ID, but are we looking for Uniprot in GFF? Okay, sorry. So this is what the file looks like. I think I talk about that here. Let's do the questions first. So what do you have as an input? You only have the FASTA file as an input. And then of course the program has embedded in it all kinds of databases that it uses. What is the output and where is it? So it's where we said it will be, in the annotation folder. And there's all kinds of information in there. So there's nucleotide FASTAs, amino acid FASTAs, log information, error information, GenBank files, GFF files. So there is a whole bunch of information here to look at. And that's every time only about one of these bins. What does it look like? A lot of FASTA files, a lot of tables, like they always are. Do you know what these output files are or mean? Well, you kind of busted me on that. I don't know what all of them mean, but I do know what some of them mean and how to use them. So we're gonna do the next part now. So like I said, very elaborate, blah, 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 blah. These Uniprot IDs. So Uniprot is a database, and they give certain unique IDs for certain proteins, and they are listed in these FASTA files, and we want to have them. So this actually we already did just above. So let's do this now, where we use grep, and then we get a Uniprot ID out. So I put in a GFF file that I would like to use, data / annotation / bin1 / what is it, prokka .gff, and let's just for fun just build up. So I'll cut this out and we'll build it up as we go. I give it a head. What we now get is all lines that have this Uniprot ID, Uniprot KB, and then some code. And this code refers back to some protein in the database. Okay, but there's a whole lot of information there, and I want to kind of clean it up. So what did I do in the original one? I used the cut function, like that, the cut function to take the part before the semicolon. So where is that? That's over here. Now let's have a look where that is. How would that look like? It would look like this. Okay. So again, oops. And then I take the part after the colon. So only that part. Let's just, for fun, see what that looks like. Remove this and look at the first 10 lines. It looks like this. So this is the file that we're getting. And we're naming this file list of Uniprot IDs. Let's name it .text. There we go. Of course, we always double check if this works. Head list of Uniprot IDs. Yes, it worked. So we have now a list where all our Uniprot IDs are. We can do word count lines of this file. There's, for this one bin, there's actually 2072 proteins that have a Uniprot ID. So those are actually not all proteins in this selection. We do lose something. There's nicer ways to do this, but this is the quickest. So we have this list of Uniprot IDs, and now we're gonna follow instructions again. Okay, prokka gives you Uniprot IDs, but actually I now want to have KO IDs, and these are the what is it? Kyoto Encyclopedia of Genes Ontologies IDs, KO IDs. So Uniprot actually has its own converter of stuff. So first I need to download my little list of Uniprot IDs in the files. Is it here? It has, yep, there it is. Click this and download. Download. Only takes a second. Where was I? I'm not going to upload a file here, the file that I just downloaded. The Uniprot IDs, there we are. Push that in. I'll enlarge this a bit for those of you with smaller screens. So the files is there. It's hardly readable, but it is there. We're converting from Uniprot KB, so we saw this, to the KO. It's somewhere down. Okay, come on. There we are. To KO. Submit. Takes a couple of seconds. And there we have it. So this is the Uniprot ID we put in. And these are the KO IDs that come out. So just the K IDs that come out. So there's again a whole bunch of them, but do note again that we lose a part. So really this is the quick and dirty method that will allow you to see your results quickly. Let's download this, the mapping table. I'm clicking mapping table here. For me it pops up actually not as a download but just on the web page. That's fine for me. And I copy this, Ctrl A, Ctrl C, copy everything. back to the instructions. Make sure to download the mapping table, step 2. It's up here. Let's correct that right away. The mapping table, and we're gonna put this. What's this? No, I don't like that one. I want this one. So we're gonna put this in the Kyoto Encyclopedia of Genes and Genomes. I don't know. Not important enough now. Okay, so I go to the link over here, Kyoto Encyclopedia of Genes. You go to the web page, and I paste in the IDs, the 2 columns of tables that we just got. I think it has to be the 2 columns. And now I know it has to be the 2 columns, and I click execute. In the meantime, this takes a second, so I Google something. Let's go to google.com. File play in the pocket. Okay, in the meantime, we've got so the web page, the KEGH website is done, and we have here. I will make this bigger again. So we have here a list of certain categories of metabolism. There is a whole lot in here. So let's click a random one for a moment. It has to load. I have to make it a bit smaller again. So what's happening here? So this is an overview of metabolism. Overview of all kinds of possible metabolic pathways. And when a square like this is green, it means that the gene to do this transaction, so it was present in the list and it's present in the bin, in the genome that we have. So this is a conversion from glycine to serine. And what do we have here? We have L3-cyanoalanine, which can be converted to gamma-glutamyl or beta-cyanoalanine. So if you're into metabolism or you have a collaborator that knows something about this, then they can help you understand this better. Let's go back to this tab, clean up a bit. So I'm particularly interested in nitrogen metabolism. So there we go. Okay, there we have it. So this is an overview of possible nitrogen metabolism that can happen inside a cell. And what's green is the genes that are present. So what this bacterium can do is move nitrate from outside the cell to inside the cell, and convert nitrate to nitrite, and I think the energy comes free in that step. So this is something that the cell can get some ATP from. What's happening here? CO2 to carbonate. Okay, that's funny. And what's happening here? Ammonia, which is inorganic nitrogen, can be converted to glutamine, to glutamate, and back, etc. So this is how this cell can at least deal with inorganic nitrogen in its environment, ammonia, and convert it to organic nitrogen in the form of amino acids. And that's an answer that you can get if you're interested in nitrogen metabolism. And what's interesting to do perhaps is to do this for all the bins you have, since you assume that these are together in one tiny ecosystem. You can study the pathways that they have in common, or perhaps the pathways, how they can supplement each other in these pathways. So for example, what I see here is you can go from nitrate out the cell, in the cell, to nitrite, but you can't go from nitrite to ammonia. So perhaps the genes required for this are in the fraction of genes that we lost in the process. Could very well be. Perhaps the step to go from nitrite to ammonia is actually done by another bacterium. So that's actually quite common for bacteria, that they supplement each other's pathways in that way. could be. So in the introduction there was a link to this paper. This is the paper that we use the data of, and here we have done something similar. Yes, there we are. So this is also the nitrogen metabolism, and here we've studied the nitrogen metabolism for a couple of the bacteria that we found in here. And we found indeed such a thing, nitrate to nitrate extracellular to intracellular to nitrite. There's bacteria that can do the step from nitrite to ammonia, and from ammonia to glutamine. So that's pretty much it for that part. So what's the lessons you can learn from this? You've now successfully completed the practical. You have taken a metagenome assembly. You've done some genomic step on this. You did some mapping, back mapping, sorting of BAM files. You know what binning is, and you've done this. You've used sequence composition binning signals, as well as differential abundant binning signals. You have checked if the bins that you got from this process have okay quality, yes or no, and then you went a step further and you annotated all genes present in these bins, and you now have a basic understanding of how you can take those genes and plot them in a metabolism pathway plotter. So I'll admit these are just the basics, and in a way perhaps you realize, especially when looking at pathways like these, that actually the workflow that we did today perhaps is the easy part. There's a clear beginning, it's a clear end, it either works or it doesn't. The hard part actually is when you have all the data and you need to have a hypothesis or a question up front that you can answer with that data. So let's say you're interested in a niche and you want to know if and what bacteria do conversion from A to B. Then you can do metagenomics to find out what organisms are there that do that step. But without a specific question, if you just start diving into the data, there's so much after such a sequencing experiment. It's way too much to just look at and see. So you need specific questions, and the workflow actually is perhaps the easy part in a way. I hope you enjoyed the practical. I guess this is the last video, and we'll I know we have a central closing down session of the practical on Friday. So be sure to join the contact hours on Friday, even if it is in some kind of video conferencing call. See you then. Thanks for doing the practical. I hope you enjoyed it, and yeah, if there's any questions, you know where to find us in the Microsoft Teams thingy where you can talk to us. Thanks.